Naktuinbouw Laboratories uses various techniques to determine whether propagating material is free of plant pathogens. These tests focus on the detection and identification of pathogens like bacteria, fungi and viruses and are performed on large scale. A number of techniques will be explained. Propagation material can be tested rapidly and with relatively low costs for the presence of pathogens using detection on semi-selective media. This method is predominantly applied in the detection of fungi and bacteria in seeds. The material or an extract of the material is distributed on a specific growth medium. Antibiotics are added to the growth medium to make it semi-selective. The antibiotics reduces the growth of undesirable bacteria, so-called saprophytic bacteria. This allows the target pathogen you want to detect to multiply unhindered. After a specific period of time, under controlled conditions in an incubator, the medium is inspected visually for the presence of pathogenic fungi or bacteria. Detection is based on morphological characteristics such as shape or color and can be conducted by the naked eye or using a microscope. The identity of suspect bacteria can be confirmed using a complementary technique, for example, immunofluorescence or PCR. ELISA is often used to test plant material. ELISA stands for Enzyme-Linked Immunosorbent Assay. The principle of ELISA is the interaction between the protein of the target pathogen and an added antibody. Plant material is ground and the plant sap is collected in tubes. The microtiter plate is coated with a specific antibody and the sample is added to the well. If present, the target pathogen binds to the antibody. Subsequently, the plate is rinsed to remove unbound traces of the sample. A second antibody is added, which is linked to an enzyme. The second antibody also binds to the previously bound pathogen. After rinsing the plate again to remove unbound antibodies, a chemical is added which is converted into a measurable yellow pigment by the enzyme. This color change reaction can be measured using a photometer. The amount of yellow pigment is a measure of the amount of pathogen present. The PCR method is applied on a large scale at Naktuinbouw. This polymerase chain reaction is more sensitive than ELISA. It allows detection and identification of low concentration levels of pathogens in seeds, plant material, soil and water. The PCR amplifies a specific piece of DNA from the target organism until there is sufficient to allow analysis. To conduct the PCR, two primers are selected that match a complementary DNA sequence of the target pathogen. The right combination of primers is specific to the pathogen you wish to identify. DNA is isolated from the sample and heated so that the DNA double helix melts and separates into two strains. Each primer binds to the complementary DNA sequence of one of the two DNA strains. Subsequently, the enzyme DNA tag polymerase extends the DNA fragment by coupling bases to the primer. After this extension, the DNA is heated again and the process is repeated. Not only the original DNA strain, but also the newly formed DNA fragments are used for amplification. In this way, the specific DNA fragments are multiplied exponentially. By repeating this process 30 to 40 times, sufficient DNA fragments are generated for analysis. With the conventional PCR method, the multiplied DNA is loaded onto a gel and fragments are separated on size. After separation and staining, the presence of specific DNA fragments can be determined. Where the real-time PCR technique is applied, a fluorescent signal is released during the process of primer extension. The PCR reaction can be followed in real time by measuring the fluorescence levels. The real-time PCR technique is more sensitive and provides more information than the conventional PCR. Bioassays are used in the detection of alive, unknown pathogens. The main difference with other tests applied by Naktuinbouw is that bioassays are conducted on plants. Tests are therefore carried out in the field or in a greenhouse and not in a laboratory. A large proportion of the viruses that are present in fruit and ornamental crops can be detected by using biological methods. Herbaceous indicator plants, also called test plants, are inoculated with plant sap derived from the plant to be tested. This is called sap inoculation. When pathogens are present in the sample, the test plant will show either local or systemic symptoms within several weeks. Apart from herbaceous indicator plants, woody indicator plants are also used. A large percentage of virus diseases in fruit crops can be detected by field testing. Using various grafting methods, tissue of the plant be tested is transferred to virus-sensitive indicator plants. The indicator plant is inspected for the presence of symptoms at various stages. For some pathogens, it can take several years before symptoms appear. Field testing is essential to detect viruses such as rubbery wood in apple and pear. Bioassays for soft fruits such as strawberry and raspberry are carried out on test plants from the same family. After leaves have been grafted onto test plants, the symptoms become visible after 2 to 8 weeks, depending on the virus species. Apart from performing various tests, Naktuinbouw continually improves existing testing methods and develops new ones. They also ensure that new and often expensive scientific tests and developments are translated into reliable and affordable tests, which can be applied on a large scale. As a result, Naktuinbouw hereby contributes to high quality and healthy horticultural propagation material. Naktuinbouw, quality in horticulture.